Bismillahirrahmanirrahim, and we start now the third video on June 22, question paper 41. Now this is the third video. We'll start with question number 8. Over the past 200 years, many species of animals and plants have become extinct. Figure 8.1 shows the changes between the year 1800 and 2000 in the number of species becoming extinct and the size of the world human population. So on one side we have the world human population at 10 raised to the power 6 and the number of known extinct species. So you can see as time is going on what is happening. The key you can see is the number of species, so let me give it a different color. So this is the number of species is stable, then it starts to number of species, known extinct species. They have risen from 1950 onwards. They weren't a lot in 1800, then 1850, same in 1900. And they started to increase from 1900 to 1950. And then from 1950 to 2000, from something like 5,000, 6,000, 7,000. Well, of course, if it's 10,000, 5,000. So from 7,000 they've gone up to, you see, you're reading it on this side. From 7,000 they've gone up to nearly 30,000. And then you look at the world human population. So let's give that another color. The human population is now this one. So human population has increased a bit, then increased more, then more in 1900 to 1950. And then from 1950 it's really shot up. It's 5,000 something into 10 raised to the power 6 world human population. The first part of the question says it has been suggested there is a correlation between the number of species becoming extinct and the size of the world human population. Suggest reasons for this possible correlation. Why more extinct as people are increasing? It's simple: habitat loss. Why are we cutting down trees and forests? For agriculture, for building roads, forestries or mines. We are hunting or fishing. We are collecting plants. We humans are spreading animal and plant diseases. We humans are spreading and bringing in alien invasive species. And of course, we are very good in polluting. We're using pesticides and polluting the environment. So habitat loss is also resulting in extinction. Then why are we doing habitat loss? Because we're clearing up forests for agriculture or for building buildings or for having roads, or the human beings are hunting and fishing animals, and that is of course resulting in depletion of their young ones, so there's nothing left to breed and slowly they become extinct over a period of many years. Then we collect plants, then the human promotes the spread of diseases, like we bring in some plants from another country and then we just we don't know what we're bringing diseases with it. Then the humans introduce alien species. We bring in a new type of cactus, and that cactus spreads so much that now there's no way to control it. Then there's pollution. You could be polluting. There could be result of overuse of fertilizers by human beings to increase crop yield as the human populations are increasing. So more and more fertilizer means more and more effects of eutrophication will be seen. Part 2, calculate the rate of species extinction per year between 1950 and 2000 and show your working. Rate of species extinction. Now, how would we calculate that? So as you can see, from 1950 to 2000, so in 2000 it was 29,000. You have to read off this side, number of known extinct species, and in 1950 it was 7,000. So 29,000 minus 7,000 over 2000 to 1950 is 50 years. So divide this by 50. This is now 22,000 divided by 50, and this is equal to 440. So 29,000 minus 7,000 is 22,000, and 2,000 minus 1950 is 50, so divide that by 50 is equal to 440. So this will get you your 2 marks. B. Extinction of animal and plant species reduces biodiversity. Explain why it is important to maintain biodiversity. Again, this is a syllabus point, so it's not something new, and you can see it is for 7 marks, so there's a lot of marks for this sort of a question. So it's just like the previous essay questions we used to have in the good old days. You see, now in these sort of questions, you should write more, because in order to get your 7 marks, you have to say, so any 7 from what? If you said a named food, it's important to maintain biodiversity because a plant provides us food. Then some plants are important and they have medicinal value. Then from plants we can get wood and fiber and paper or rubber. Then genetic diversity for future use. Maybe we need those plants which are now maybe 100 years from now, or maybe those plants which were there 100 years before, they had some very good genetic information. They had some very good alleles which caused them to be very strong during years and years of droughts. Then we can do some research on them. Then of course education, because it's important as education when children or we as human beings do research and we have information about many plants and animals which have now become extinct. We could easily have used them for a better research and finding out the cure for certain diseases. Then aesthetics. Nature is beautiful. I mean, I love walks in nature, very nature parks, and we go to Swat and we go to the northern areas to go and see all these beautiful places. Then tourism. This when people visit all these places, there's a lot of economy being generated. People visit northern areas. They live in a hotel. They do some shopping. They eat. So a lot of people's economic needs are fulfilled. Then ethical and moral issue, yeah, we must leave the world as we found it for future generations. Then local cultural significance. Then they maintain food chains and food webs. Then pollinators, we have, if they're insects, they're going to be pollinators and they will remain balance the ecosystem. Then plants will protect against soil erosion. Then the soil formation takes place when plants and animals die and all the nutrients go back into the soil. And you can talk of named cycles like the nitrogen cycle, which helps in recycling the nitrogen content. And then climate stability, more trees, more transpiration, more clouds, more rain, and keeps the earth cooler. Now coming to question number 9, A. Stomata are involved in both transpiration and photosynthesis in plants. Figure 9.1 is a diagram of an open stoma, its guard cells and surrounding epidermal cells. Figure 9.1. Complete table 9.1 by choosing the correct letter from figure 9.1 to match the feature stated in table 9.1. Okay, each letter may be used once, more than once, or not at all. Location of the Calvin cycle. Location of the Calvin cycle, and made mainly of cellulose. Now we know location of the Calvin cycle would be B. Why would it be B? Because you see, these are the chloroplasts. These things are the chloroplasts, and the inside of it is the stroma of the chloroplast. And that is why it would be this, see, there was a little confusing. Why? Because there was another label which was the A. Now what was A? It was the nucleus. The nucleus of the guard cell. So there are nucleuses everywhere. There are 4 nucleuses which are shown, 2 of the guard cell and 2 of the epidermal cells. So location of the Calvin cycle and made mainly of cellulose is the cell wall, and that was D. So the cell wall is the inner wall, which of course the inner and the outer wall all is cellulose of the guard cell. So that answer was D. Then coming to the B part of the question, the opening and closing of stomata are due to changes in environmental conditions. Hydrogen ions and potassium ions are involved in the opening of the stomata. Describe how hydrogen ions and potassium ions are involved in the opening of the stomata. Well, I think if you have looked at my video on this, it's very, very clear. Hydrogen ions are pumped out of the guard cell. So these are the guard cells. So hydrogen ions are pumped out of the guard cells. Then inside of the cell becomes negatively charged. It becomes negatively charged naturally because you pumped out all the positive ions. So potassium starts to enter by, why? Because it's a phospholipid bilayer. So potassium starts to enter by facilitated diffusion. And when a lot of potassium enters the cell, what does this do? It lowers the water potential of the cell, decreases. So water potential of the cell decreases. When the water potential of the cell decreases, what is going to happen? Water enters by osmosis through the aquaporins. So water enters by osmosis. Now what is this going to happen? This is going to make the cell expand in volume, and then it's going to pull this inner one like this. So water enters the cell by osmosis, cell expands or increases in volume, and the stoma opens due to the thick inner wall. This is the thick inner wall. This is going to be this thick inner wall this is thicker. It's thicker than the rest of the wall. So that is pulled outward, and this stomata opens. So H positive pumped out of guard cell, which I just showed you here. H positive pumped out of guard cell, then inside of cell becomes negatively charged. Then potassium enters by facilitated diffusion, water potential of the cell decreases, water enters the cell by osmosis, cell increases in volume slash expands, stoma opens due to thick inner wall. Now at last we've come to the last question. This was a 2-hour paper, so pretty lengthy. A part, motor neurons and sensory neurons have different roles in nervous coordination. Contrast the structure and function of the sensory neurons and the motor. Contrast means you would give what is different about each of them. So the motor neurons transmit impulse from CNS to effector. Sensory transmits impulse from receptors, sense organs to the CNS. So this was the biggest contrast that we had. And then any two from cell body at the end of the neuron, cell body in the middle, then cell body in the CNS, cell body in the dorsal root ganglion, then long axon, short axon, and dendrite attached to the cell body, and dendrites attached to the dendron. So 3 marks, and you could have done any of these, and you could have got your 3 out of 3. So you could have easily done also a table with giving the contrasting features. So transmits impulse from CNS, transmits impulse from receptors to CNS. The other was CNS to effector. Okay, so we talk about this. I'm just going to give it to you how you write it. So whenever it says a contrast or a compare is sort of similarities and differences, but a contrast means only very, very similar things like differences. Like if you were comparing two students, I would say, oh, so and so wears glasses and so and so does not wear glasses. So in the motor neuron is CNS to effector, receptor to CNS. In the motor neuron, the cell body is at the end of the neuron. In this, the cell body is not at the end of the neuron. That's a very good way of saying it if you're not even very sure if you have to write it is in the middle or anything like this. Cell body in the CNS, cell body in the ganglion, then long axon, short axon, and dendrites attached to the cell body and dendrites attached to the dendron. The B part of the question. Mammals have many types of neurons which vary in axon diameter and myelination. Some are myelinated, some are not myelinated, and the axon diameter varies. Table 10.1 shows the axon diameter and the mean impulse transmission speed of 4 different types of mammalian neurons. So alpha, beta, gamma, and motor C. Myelination present in the first three, this, and not in this one. Then mean axon diameter was 13 to 20, 6 to 12, 1 to 5, and zero point 0.2 to 1.5. And the mean impulse transmission, you see, as you can see here, the more the diameter, the more the mean impulse transmission speed is. And as the diameter is decreasing, so is this decreasing. So you can see this, you can compare this with this. And you can see that the one with the myelin are all between 5 and 120. And the motor C is the one which has the least transmission, does not have any myelin, and it is 0.5 to 2. So with myelin, it was all above 5, and well, it's even less than 2, 0.5 to 2 in the one which does not have any myelin. And so you're going to see this here, no myelin here. Using the data shown in table 10.1, comment on the relationship between myelination and mean impulse transmission speed and axon diameter and mean impulse transmission speed. Now that was very easy. It wasn't a very difficult question. If you look at this, what is it? No myelin. So myelin increases speed. You can say no myelin, slow, and with myelin is faster, or you can say myelin increases speed. And the second one was no myelin is zero point 0.5 to 2 meters per second, and with myelin is anything above 5 up to 120 meters per second. You see, from 5 up to 120 meters per second. As axon diameter increases, speed increases, and then you could give me the motor alpha and speed for two neurons, and motor C. So you could have given me the diameter. So the diameter was 13 to 20 micrometer. You must give me the units. And the speed was in meters per second. So if you gave me figures from motor alpha and this, and you gave me their diameters and their speeds and the diameter and the speeds, any two neurons, I'm saying you needed to compare two so that you see this is called when you give some data that gives you some marks. So that would, if you just wrote those down, you got a mark for that as well. Because why did you have to give that? Because they say data shown and comment. So using the data shown, when they say using the data, then you must use the data. Using the data means you must use the data. That's a bit of a joke. So you can see myelin increases speed, no myelin 0.5 to 2 meters, and with myelin more than 5 up to 120 meters per second. This is one another mark. Axon diameter increases, so speed increases, and then of course you gave me the data figures for any two neurons. I've compared motor alpha with motor C, but you can do any one. You could have done B and gamma or delta, whatever that sign is. The end of the question, this is the last part of the question of the paper. In fact, figure 10.1 is a graph of an action potential in a mammalian neuron. Figure 10.1, you're seeing A, B, C. This is the membrane potential in millivolts, and this is the time in milliseconds. Now, what are they asking you? They're asking you a difficult question. They're saying, with reference to figure 10.1, suggest why no further action potential can occur during A and B. So they want to know why during A and B no action potential can occur. Now that's of course the refractory period, but why they're asking you, they're not asking you the name for that, they're asking you why that cannot happen. And with reference suggest why no further action potential can occur during A and B, and it is difficult for the further action potential to occur during C. Now during C there is hyperpolarization. So you say from minus 70 it's gone down to minus 85, in fact more than 85. So now they're asking you explanation for that. That's a difficult question. I must say that's a very difficult question. So why, and it is only 3 marks. And that's what a total of 9 marks was this question. So what is happening in these for 3 marks? What do you give me? Now at A, what is happening? Now if you remember the mnemonic that I've given you is Ni, sodium in, and this is Ke, potassium exit. So this is called depolarization. During depolarization, what is happening? Sodium channels are open and sodium ions are diffusing in. So already sodium ions are diffusing in. How can a further action potential be fired? Action potential is sodium ions are already flowing in. So at A, why cannot be an action potential? Because sodium ions are already open and sodium is already entered the neuron. At B, why can another action potential not be fired? Because at B, the sodium channels are inactive. In fact, the potassium channels are open, and that is why the potassium ions are exiting. The potassium channels are open and the potassium channels are exiting. Sodium channels are not responsive, they are inactive, unresponsive. and the membrane is impermeable to sodium. You see, depolarization can only occur if sodium ions enter. Now at C, why can further action potential cannot be fired at C? Now C was this part. C was this part. Why is it? Because it's harder to reach the threshold. Or you can say the potassium channels are still open, and sodium potassium channel needs to restore the action potential, sorry, the resting potential. Or you can say there is hyperpolarization at C. So there were very distinct 3 mark scheme points which you had to give me for A, B, and C to get your 3 marks. So this would be the very simple way of wording it, that sodium channels are already open at A, so depolarization or reaction potential cannot occur. And at B, sodium channels are inactive. In fact, the potassium channels are open, so that is why it won't happen. And in C, it is harder. But it says it's difficult for further action potentials to occur during C. Why is it difficult? Why is it difficult if it's harder to reach the threshold? If the threshold is harder to reach, then how can an action potential be fired? An action potential is only fired when the threshold is crossed. And why? Because at C, there is hyperpolarization at C. So then you would need a very strong stimulus to cause the threshold to be crossed and an action potential to be fired. And that completes your paper 4.1 for this year and this June 22 paper. And I hope this is all very helpful, and I hope this is all going to help you revise your paper 4s and do well in the paper 4s in the forthcoming exams.